genetic Chiari malformations.
Finocchiaro lavora all'Istituto Neurologico, si interessa di ricerca e soprattutto di neuro-oncologia.
Genere si ringrazia per l'invito. In realtà
I would like to thank you for the kind invitation, even though I went through a lot of sufferings and pain to put together this presentation. It was very painful for me because I'll try to give you some useful information, but it is far from easy to summarize the genetic abnormalities in the Chiari malformation. In any case, I'd like to start by telling you what we can rely upon to say that we have this genetic component. Well, to define a genetic trait, we need a few elements. First of all, we have to document familial aggregation, first of all, or clustering twin studies, or we need to have statistically significant segregation analysis, meaning that every time there is a genetic alteration, we look for statistical significance, or an increased risk for relatives of affected individuals compared with the risk of for the general population. We have seen that prevalence accounts for 0.7%. And then now we have to see whether there is a co-segregation of the condition with other known genetic traits. Some 8 years ago, some interesting findings were reported by Spear and co-workers on a study made on 31 pedigrees in which 2 or more individuals were affected by Chiari type 1. And if we take a look at this table, we can see that some of the conditions we mentioned before are met, because we have 78 individuals with this disease, with prevalence among female subjects, though it is not statistically significant. And then we have an evidence of families showing a vertical transmission of the disease, and then the evidence of siblings affected and cousins as well. And therefore, and also here we see that 21% of first-degree relatives of affected patients were diagnosed with Chiari malformation associated with syringomyelia, where syringomyelia was present. So all in all, these families meet a few of the criteria that I showed before. And also, 16 syndromes or genetic disease in which the familial component can be associated with Chiari malformation. So the first limit was set by this study that proved that at least one subgroup of patients with Chiari has a familial component. But nothing was said about what gene or genes were involved. As it was illustrated before about the development of the nervous system, in the Chiari malformation we primarily have a mesodermal disorder involving the basicranium and the craniovertebral junction. This is a complex process that depends or that is based on somites, and that depends on the expression of some key genes for the development of the nervous system and of the mesoderm. I'd like to focus on two genes in particular. One is PAX1, which is key for somatic segmentation and for the differentiation of the structures. You can see here, I don't know if you can see, in any case, the in situ hybridation studies on mouse embryos. In this case, PAX1, which is an homeobox gene related to some DNA regions, hybridizes in the regions which are key for the development of regions involved, affected in the Chiari malformation. The same can be said for OX genes. This is a very well preserved family of genes located in the genome, as you can see with these arrows, that is present in many different organisms. And as far as the OX family is concerned, I can tell you that recent studies are in any case far from understanding the target genes for these homeoboxes. So this is still unclear. However, there is no evidence that PAX or OX are mutated or significantly altered in the Chiari disease. And so this study posited a few assumptions, however, that were not proven in the end. In any case, the goal is to study large families, large pedigrees, large number of pedigrees. This doesn't only apply to human beings, but also to animals, because by observing a large number of pedigrees of this kind, we can obtain useful data. In the case of this nice little dog, the Cavalier Chinchilla Spaniel, the presence of an anomaly was identified in the occipital bone, an anomaly that is very similar to the that of the Chiari malformation. It was studied in over 5,000 animals. All in all, they were studied through 24 generations with 100 cases reported. And in this study, two, let's say, culprits were identified, but once again, from the genetical standpoint, we did not understand exactly which was the gene or which were the genes involved. Moving to the human beings, in 2006, the findings of a large cooperative study was published by Duke University, a study that involved 23 families. Here you see the pedigrees with 71 individuals affected by Chiari and neurologically diagnosed based on the criteria we mentioned before, using 10,000 SNPs, single nucleotide polymorphisms. This study started to give us a few interesting ideas about the genetic component of the Chiari malformation. Before discussing this, I'd like to tell you what the SNPs are. These single nucleotide polymorphisms are often present in our genome, they contribute, even though they are not in the codifying part of the gene. You know that only 3% of the genome codifies for gene, and we do not know yet about the role of the rest, the remaining 97%, which for the time being is called junk DNA. But in any case, the presence of these polymorphisms can help us carry out segregation studies, meaning that if in a certain family 12 or 50 polymorphisms are the same between two affected individuals, we can reasonably assume that in this region there is something involved in that specific disease we are investigating, for instance, in the Chiari syndrome. And with the various statistical analysis, the 9 and the 15 chromosome, the 9th and the 16th chromosome were investigated. We have the fibrillin 1 in chromosome 15. Here you see an outline of this structure. This calcium binding modules are present in the C-terminal, in the C-terminal end, and you see that chelating calcium the calcium chelation can modify dramatically the structure of this protein. Well, fibrillin has is already involved in the pathogenesis of a very well-known disease, the Marfan syndrome, the connective tissue disease with various disorders affecting various systems, and also apparently President Lincoln was affected by this syndrome. But once again, I haven't found any data supporting the presence of the mutation of fibrillin 1 in Chiari patients. The mutation right now is only one of the possibilities for a gene alteration, because a gene can become dormant for various with various techniques and with various systems. So we are at a time when there are probably new discoveries around the corner, but in any case, this is what we know about the genetic component of the Chiari disease so far. What is well defined is the presence of mutation of the receptor for the growth factor, a receptor, type 2 receptor, in patients having genetic syndromes associated with Chiari and syringomyelia. This is the result of a small cohort of patients in Japan affected by Crouzon syndrome, which is a dominant disease involving craniofacial alterations, hypotaylorism, and apoptosis and hypoplasia of the central portion of the face. Here you see these images, and you see Chiari that can be associated also with syringomyelia. In the FGFR2 receptor, we find, in these 5 cases, mutations in key portions of the protein structure, because they cancel out cysteines, which are very important residues for this structure. And here you see a few findings, and here you see the mutations that were identified. The FGFR1 mutations were also found, and here you see some raw data. These are the sequence profiles showing the mutations that were first identified with the single-strand conformation polymorphism technique. Another FGFR2 mutation was found in a patient with Chiari-like malformations of the anterior chamber of the eye. A few weeks back, also another case was published, again in Asia, with a rare syndrome called Beer-Stevenson syndrome, with an FGFR2 mutation, always involving cysteine. And therefore, in various cases associated with Chiari, we find these mutations. And so this is perhaps one of the key messages that we have to the key lessons we can learn from my presentation. And then the last very interesting finding, also very, very recent, refers to the mutation of another gene of belonging to the same family of the genes controlling development, OTX2, which appears to be mutated in a Japanese patient who has delayed development with a severe pituitary deficit associated with Chiari malformation. In this case, the mutated protein remains in the nucleus, but the mutation prevents it from performing its physiological role, that is to say, activating other genes, such as these two indicated here. Here you see the structure of the protein, and you see that the insertion of these two nucleotides causes this anarchy of the DNA sequence downward. OTX2 is part of a highly conserved family of homeodomain containing transcription factors that are present in many genes. Here you see these genes upward regulated or repressed, depending on whether you have a light blue or a red triangle. This is not very relevant to us, but I'd like to remind you that at least in this case, a connection was identified between a gene regulating the development of this region and this type of malformation. Therefore, in conclusion, the take-home messages I can offer you, hoping that the future will give us something more, are four. There is a genetic basis in a subgroup of CM patients. An interesting finding is that a study on 20 pedigrees or 20 families with dozens of affected patients has identified two candidate regions with alterations that could be associated with Chiari. In one, there is let's say a plausible gene, fibrillin 1, and then in the 9 chromosome arm, no candidates have been identified yet. And then several reports suggest that the FGFR2 is a candidate CM gene, and also, last but not least, it is very important to work in large studies on large samples, because this can be very important to identify this site of the syndrome. Thank you very much for your kind attention.